Lyme disease is a vector-borne disease caused by the Borrelia bacterium, which is spread by ticks in the genus Ixodes. Lyme disease is caused by spirochete spiral bacteria in the genus Borrelia. No human vaccines for Lyme disease are currently available. The only human vaccine to advance to market was Limerick's, which was available starting in 1998 before being pulled from the market in 2002. The vaccine candidate VLA-15 was scheduled to start a phase 3 trial in the third quarter of 2022, and research is still ongoing. Multiple vaccines are available for the prevention of Lyme disease in dogs. Lyme disease is the most common tick-borne disease in North America and Europe, and one of the fastest-growing infectious diseases in the United States. Of cases reported to the United States CDC, the ratio of Lyme disease infection is 7.9 cases for every 100,000 people. In the 10 states where Lyme disease is most prevalent, the average was 31.6 cases for every 100,000 persons for the year 2005. It is important to make a distinction between the borreliosis bacteria and the full spectrum disease known as Lyme disease. Lyme disease was diagnosed as a separate condition for the first time in 1975 in Lyme, Connecticut. Outside of the US and English-speaking world, the disease is known simply as borreliosis. The evolutionary history of Borrelia burgdorferi genetics has been the subject of many recent studies. Perhaps the first detailed description of what is now known as Lyme disease appeared in the writings of John Walker after a visit to the island of Jura, Deer Island, off the west coast of Scotland in 1764. He gives a good description of both the symptoms of Lyme disease with exquisite pain in the interior parts of the limbs and of the tick vector itself, which he describes as a worm with a body that is of reddish color and a row of feet that penetrates the skin. Many people from this area emigrated to North America between 1717 and the end of the 18th century. The modern history of medical understanding of the disease, including its cause, diagnosis, and treatment, has been difficult. The full syndrome, now known as Lyme disease, was not recognized until a cluster of cases originally thought to be juvenile rheumatoid arthritis was identified in 3 towns in southeastern Connecticut in 1975, including the towns of Lyme and Old Lyme, which gave the disease its popular name. This was investigated by physicians David Sneidman and Alan Steer of the Epidemic Intelligence Service, and by others from Yale University, including Stephen Malowitza, who is credited as the co-discoverer of the disease. The recognition that the people in the United States had EM led to the recognition that Lyme arthritis was one manifestation of the same tick-borne condition known in Europe. Before 1976, elements of B. burgdorferi sensu lato infection were called or known as tick-borne meningopolyneuritis, Guerin-Bujodow syndrome, Bannewarth syndrome, Afzelius disease, Montagny, or sheep tick fever. Since 1976, the disease is most often referred to as Lyme disease, Lyme borreliosis, or simply borreliosis. Plum Island. Plum Island is an island in the town of Southold in Suffolk County, New York. The island is situated in Gardiner's Bay, east of Orient Point, off the southeastern end of North Fork coast of Long Island. It is about 3 miles long and 1 mile wide at its widest point. Only 17 miles south of Lyme, Connecticut, is the island site of the Plum Island Animal Disease Center, PIADC, which was established by the United States Department of Agriculture, USDA, in 1954. The island is also the site of the former U.S. military installation Fort Terry and the historic Plum Island Light and its automated replacement. In 2003, the United States Department of Homeland Security assumed ownership of the island and its facilities. The USDA continues its long-running science mission at PIADC jointly with DHS, who are charged with the safe and secure operation of the facility. As one result of the heightened national security initiatives following the 9/11 attacks, Plum Island was considered as a potential site for a new high-security animal disease lab, National Biological and Agro Defense Facility, NBAF. In September 2008, the U.S. Congress passed H.R. 2638 that directed the General Services Administration to close the PIADC, sell the island to the public, and use the proceeds toward the construction of the NBAF if it were decided that the NBAF would be built elsewhere. In January 2009, the United States Department of Homeland Security selected the city of Manhattan, Kansas, as the site for the new NBAF and decided to relocate the PIADC there as well. However, the decline in real estate values stemming from the subprime mortgage crisis in the late 2000s recession caused the sale of the island to be considered no longer viable. Because the proceeds from selling the island were needed to construct the new facility in Kansas, the project was effectively canceled for the short term when the 2013 federal budget request contained no further funding for the facility. As part of the planned sale of Plum Island, the United States government prepared an environmental impact statement for the island, one objective of which was to determine whether the impact of nearly 60 years of animal testing on the island constituted a threat to the American public health, that could preclude the planned sale. In 2008, U.S. Congress passed and then U.S. President George W. Bush signed H.R. 2638, part of the Consolidated Disaster Assistance and Continuing Appropriations Act 2009, which required that Plum Island and the Plum Island Animal Disease Center (PIADC) be sold to help fund the new excuse me, the new biological and agro defense facility in Manhattan, Kansas. This legislation started the process that would have had all of Plum Island be sold to a private party. A regional environmental group, Save the Sound, led the legal aspects of the movement to preserve Plum Island, including the repeal of H.R. 2638. In addition, a coalition of conservation, environmental, and civic organizations formed the Preserve Plum Island Coalition, PPIC. The PPIC advocates a plan for its designation as a national wildlife refuge or other protected area. The proposal provides for guided public access to the historic district in a trail area where nature preserve areas at the south and east would be restricted to student and research access. On July 16, 2013, former U.S. Congressman Tim Bishop introduced a new bipartisan bill, Save, Don't Sell Plum Island. This goal of the bill, the goal of this bill was to preserve critical biodiversity and prevent further development on Plum Island by eliminating H.R. 2638's provision that all of Plum Island be sold at public auction to fund the new NBAF in Manhattan, Kansas. Similar legislation was also introduced in the U.S. Senate at the same time as that in the U.S. House of Representatives. On August 29, 2013, the USDA and DHS announced a final record of division which determined that the entirety of Plum Island was to be sold to a private sector to help offset the cost of constructing the new replacement facility. On September 30, 2013, New York Governor Andrew M. Cuomo sent a letter to the United States Department of Homeland Security and the United States General Services Administration calling for both agencies to submit to a consent order requiring them to present a comprehensive environmental cleanup plan for Plum Island in Suffolk County, New York, and giving New York State final review of the island's conditions before it would be put up for sale. On January 5, 2015, the Connecticut Fund for the Environment, CFE, and Save the Sound jointly sent a letter to the U.S. Department of Homeland Security and U.S. General Services Administration indicating a 60-day notice of intent to sue these agencies. This letter outlined how the sale of Plum Island under the terms of H.R. 2638 was in violation of the Endangered Species Act. On April 1, 2015, New York and Connecticut Senators, Senator Schumer, Gillibrand, Blumenthal, and Murphy, sent a letter to the Appropriation Committee asking for the repeal of H.R. 2638. The senators explained that the original financial reasoning to sell Plum Island was no longer valid and that it was in the best interest in the country to preserve Plum Island. Congress voted on December 21, 2020 to block the planned sale and preserve the island. Paperclip. Erich Traub, June 27, 1906 to 18 May 1985. Erich Traub was a German veterinarian, scientist, and virologist who specialized in foot and mouth disease, rinderpest, and Newcastle disease. Traub was a member of the National Socialist Motor Corps (NASKK), a Nazi motorist corps, from 1938 to 1942. He worked directly for Heinrich Himmler, head of the Schutzstaffel, the SS, as the lab chief of the Nazis' leading bioweapons facility on Rheims Island. From 1942 to 1948, Traub worked as a lab chief at the Reich Research Institute for Virus Diseases of Animals at Rheims Island, a German animal virus research institute in the Baltic Sea, now named Friedrich Loeffler Institute. The institute was headed by Professor Dr. Otto Waldmann from 1919 to 1948, while Traub was vice president. On orders from Himmler and Bjorn, the deputy Reich health leader and the head of the German biological warfare program, Traub worked on weaponizing foot and mouth disease, which has been reported to have been dispersed by aircraft onto cattle and reindeer in Russia in 1944. Bjorn sent Traub to pick up a strain of rinderpest virus in Turkey. Upon his return, this strain proved inactive, non-virulent, and therefore plans for a rinderpest product were shelved. Immediately after the war, Traub was trapped in the Soviet zone of Allied-occupied Germany. He was forced to work for the Soviets from his lab on Rheims Island. In July 1948, the British evacuated Erich Traub from Rheims Island as a high-priority intelligence target, since it was now the Soviet zone, and they feared that Traub would be assisting in their biological warfare program. Traub denied this, however, claiming that his only interest was foot and mouth disease in animals. Traub was brought to the United States in 1949 under the auspices of the United States government program Operation Paperclip, meant to exploit Soviet scientific knowledge in Germany and deny it to the Soviet Union. From 1949 to 1953, he was associated with the Naval Medical Research Institute in Bethesda, Maryland. Months into his Operation Paperclip contract, Traub was asked to meet with U.S. scientists from Fort Detrick, the Army's biological warfare headquarters in Frederick, Maryland. As noted, as a noted German authority on viruses, he was asked to consult on their animal disease program from a biological warfare perspective. Traub discussed work done at the Reich Research Institute for Virus Diseases on Animals on Rheims Island during World War II for the Nazis, and work done after the war for the Russians. Traub gave a detailed explanation of the secret operation at the Institute and his activities there. This information provided the groundwork for Fort Detrick's offshore germ warfare animal disease lab on Plum Island. His publicly published research from his time in the United States reports disease research not directly related to weaponization. In 1951, he published a report for the Naval Medical Research Institute on Newcastle disease virus in chicken and mammalian blood cells. 2 years later, he published a paper for the Navy on the mechanisms of immunity in chickens to Newcastle and the possible role of cellular factors. Also, in 1953, he published another paper for the Navy with Worth eye caps on the foot and mouth disease virus and methods for rapid adaptation. In theory, insects of all types, particularly the biting species, can be used as disease vectors in a biological warfare program. Germany, Japan, Britain, Russia, and the United States all conducted experiments along these lines during the Second World War, and the Japanese used such insect-borne diseases against both soldiers and civilians in China. This was one reason that President Franklin Roosevelt and Secretary of War Henry Stimson ordered the creation of an American biological warfare program in 1942, which was headquartered at Camp Detrick, Maryland. This eventually grew to a very large facility with 245 buildings and a $60 million budget, including an entomological weapons department that mass-produced flies, lice, mosquitoes as disease vectors. Although the British bioweapon facility at Portondown concentrated on the production of anthrax bombs, it also conducted experiments on insects as vectors. After the war, the Army's 406th Medical General Laboratory in Japan cooperated with former scientists from Unit 731 in experimenting with many different disease vectors, including lice, flies, mosquitoes, ticks, fleas, spiders, and beetles to carry a wide variety of diseases, from cholera to meningitis. At Fort Detrick in the late 1940s, Theodore Rosebery also rated insect vectors very highly, and its entomological division had at least three insect vectored weapons ready for use by 1950. Some of these were later tested at the Dugway Proving Grounds in Utah and allegedly used during the Korean War as well. Traub visited the Plum Island Animal Disease Research Center, PIADC, in New York on at least three occasions in the 1950s. The Plum Island facility, operated by the Department of Agriculture, conducted research on foot and mouth disease, FMD, of cattle, one of Traub's areas of expertise. Traub was offered a leading position at Plum Island in 1958, which he officially declined. It has been alleged that the United States performed bioweapons research at the Plum Island facility. Fort Terry on Plum Island was part of the U.S. Biological Warfare Program in 1944 to 46, working on veterinary testing in connection with the weaponization of brucellosis. After the war, research on biological weapons continued at Bluff in Arkansas and Fort Detrick, Maryland. While officially, at least Plum Island was transferred to the U.S. Department of Agriculture, from 1949, Plum Island also conducted work on biological weapons against animals and livestock, such as foot and mouth disease, rinderpest, Newcastle disease, African swine fever, and plague of malaria in birds. Traub's research work from the Second War onward involved at least the first three of these, only dangerous to non-human species.